Okay, hello and welcome to the second part of the lesson on meiosis. Now in this lesson we're going to go through how we manage to create variety and do our calculations. Okay, so there's a bit of maths in this as well. So the previous lesson you should know the names of the stages in meiosis, that it's two lots of cell division resulting in haploid cells. Okay, so this is the bit of syllabus. As you can see here, we're focusing very specifically on these two processes called crossing over and independent segregation or independent assortment. Okay, the idea down here as well of random fertilization of haploid gametes further increases genetic diversity. So the two parent thing really increases diversity as well. And then over there on the side you can see in the maths box, basically you need to be able to put some numbers to this. You need to be able to work out, well, how many possible combinations are there from this process of independent assortment. Okay, so in terms of prior knowledge, I'm expecting you to have watched the previous video of this. I expect you to know confidently basic chromosome and cell structure, when it's a chromosome, when it's not a chromosome, when it's doubled up chromosome and that's still one chromosome, what we mean by a pair of chromosomes, what the words homologous mean, all that kind of stuff. Mitosis, so I expect you to be familiar with the words interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and the idea from the last video that we explained that there's actually two lots of that in meiosis. So we've got a 1 or a 2 after each one. I'm also expecting you to be familiar with the idea of life cycles and where in a life cycle this happens, both anatomically within an individual and when in a life cycle. And if I'm using phrases like 1N and 2N, haploid, diploid terminology, I'm expecting you to be familiar with that as well. Okay, so cracking straight on. So here's where we finished last time in terms of our comparison. As you can see, look, meiosis has got two cell divisions in it. Okay, so we divide once and then again to form 4 cells, whereas mitosis with a T only forms 2 cells. The ones over there are identical. These ones here are not. Okay, and please notice the bits that are different, the bits I've highlighted in red here that are subject of this lesson: crossing over here and independent segregation. These are the two processes by which we generate the variety, generate the variation, which is why we need meiosis. Okay, so if you look on the other side there, where it says chromosomes line up in single file on the equator. Okay, here we can see that isn't actually happening. Okay, they're lining up, sure, but then we don't get this centromere snapping. Okay, up here as well, you can also see from up here to down here, something's changed about little sections of these chromosomes. Some of them are different colours, they look like they've swapped bits, and that's the fault of crossing over. So that's where we're going to start here. Right? The first introduction of variation, of difference between these chromosomes, is at crossing over, and that happens here, look, at prophase number 1. Now you should recognise this, the DNA is already doubled up and we've got visible chromosomes. So we've been through interphase and we're now into prophase. Right? And by this point here, right, which you should recognise as actually anaphase, where they're being pulled apart, the chromosomes look a bit different. So something's happened in the middle there at prophase 1 that's led to these little different patches having moved around, and that's crossing over, right? So moving on then, what is crossing over? Well, it happens at prophase 1, okay? And actually literally parts of the homologous chromosomes swap over with each other and you get new allele combinations, okay? So if we look at this sort of little set of lovely chromosomes down here, this pair of chromosomes, now this is a pair of homologous chromosomes, okay? Doesn't matter that they've been through DNA replication, they look like doubles, right? They are a pair. So that's one chromosome and that's one chromosome over there, okay? One chromosome, right? So unlike mitosis, where they would line up nice and neatly along the equator and get pulled apart, the homologous pairs actually line up on top of each other, right? Properly overlap physically on top of each other, forming what we call a tetrad. Okay, and what happens then is bits of the chromosome that cross over kind of twist around each other, break, and recombine in different combinations. If I show you that with my wonderful mastery of PowerPoint animation, so there they are. Look. There they formed a tetrad. Now I've only overlapped them slightly so we can see easily, but each of these places where we've got crossed over chromosomes, right, we call that a chiasma. Okay, and in this case we've got 2. There may actually be many hundreds or many thousands up or down the length of the chromosome. Okay, so what then happens is after this sort of twisting, snapping and rejoining stage is you end up with chromosomes with allele combinations that you've never actually seen before, right? They could be completely new, right? Because if this didn't happen and two genes were on the same chromosome, well, they'd always be doomed to go down the generations together. And this explains something that Darwin himself was challenged on and didn't really know very much about, which is the idea of, well, okay, how come some traits, if you're saying, traits get passed down the generations and they get inherited, but some traits seem to go together and some traits seem to work completely randomly, like Mendel's peas or this random genetic tombola, right? How do you explain that? Of course, Darwin didn't know, and it wasn't until the discovery of genes and chromosomes that we realised that things that go down the generation together, tend to go down the generation together, one, are probably on the same chromosome, and two, are probably actually quite close together physically on the same chromosome. Because if two things are on separate chromosomes, well, they'll literally move down the generations randomly. You could get one, you could get the other, doesn't matter. They're not physically attached. If they're on one end and the other end of a chromosome, as we've got here, so it might be this version up here and this this gene down here, well, a crossing over event anywhere in between them would split them up. So while it's not as likely as them being completely separate, you're going to see them splitting up occasionally. Whereas if two genes are very, very close, like right next to each other on the chromosome, you'd need a crossing over event at a very specific place right between them to split those two genes up, okay? To split those two alleles up. Now, that's not impossible, right? But it's highly unlikely, which explains why some things like pigmentation, hair colour, eye colour, skin colour, tend to go down the generations together. They can split up, you can get different combinations, of course, whereas things that are completely separate, like your hair colour and your blood type, right, go off in completely different, or your ability to make factor 8 go off down the generations in completely different ways, completely randomly, because they're different places and different chromosomes. Okay, so that's crossing over, okay, and it generates, and we've only done two events there. It could be thousands up and down, or hundreds up and down even one chromosome, let alone the 23 pairs that you've got. So that's basically what you end up with. Look, so we start with our entirely pink, and then you've got the big A and little a, big B, little b genetic nomenclature you should be familiar with. They cross over, they form that chiasma, and then you get combinations that you've never seen before. So this side here, look, that's the same as the original one, but the next one over has got dominant, dominant A, dominant B, which wasn't there originally. That is a new combination thanks to crossing over. So if we come back to our original picture, that's here, prophase 1. Now here, at anaphase, metaphase 1, we get another variety creating thing, independent segregation. Look closely here, so you now know why the colours are different, but look what hasn't snapped. We have not snapped the centromere, okay? The centromere has not broken. So whereas in mitosis with a T, if you look down at the bottom there, right, you can see that the centromere has snapped and one copy of each chromosome has gone to others. So the two chromatids have split, been upgraded to chromosomes in their own right, and gone into each cell to make identical copies. In this instance, all of one chromosome of each homologous pair has gone one way, and all of the other has gone the other way. Okay, so that's what we call independent segregation. So no snapping of the centromere. So this cell is going to end up with majority blue, majority white or cream, whereas this one's the other way around. This one could end up blue, it could end up cream. It really doesn't matter. It's random, right? It's independent segregation. Okay, so let me show you a little bit about how that works. Okay, so independent segregation is our second variety creating step. What it does, it happens at metaphase or anaphase 1. Okay, and you've got pairs of homologous chromosomes that line up on top of each other. We've already said this, the tetrads, they do their crossing over thing, lovely, but then one whole chromosome of each pair is pulled into each pole. You've not snapped the centromere and taken a chromatid each that then gets upgraded to being called a chromosome in its own right. You've taken a whole, right, all the maternal or all the paternal. Okay, so the whole chromosome is pulled to each pole. So there's no snapping of the centromere. Okay, so no snapping of the centromere. Right, so that then basically ends up with, right, this in meiosis 1. Okay, so we've got here, look, this combination lined up this way, and then over there you've got them lined up that way. So you could end up at the end of meiosis 1, okay, with this big blue, little pink. You could end up with big pink, little blue. You could end up with big pink, little pink, and big blue, little blue. It is random, okay? Then meiosis 2, we don't need to say much about meiosis 2, happens really, really quickly, Okay, happens really quickly, and all it does, no DNA replication, because we're trying to get haploid cells, right? We end up with the chromosomes lining up again. This time we do break the centromere, so we do break the centromere in meiosis 2, pulls those chromatids apart, they get upgraded to chromosomes in their own right, and then down here, look, these are all our haploid gametes, okay? So that's how we end up with 4 cells from 1, okay? And that's meiosis 2. So all of the variety creating stuff happens in meiosis 1, and then meiosis 2 is literally very quick, runs around very, very swiftly, they all line up, they get pulled apart in the same way with a metaphase, an anaphase, and a telophase, and a cytokinesis 2, and that leads us to our 4 haploid cells from 1. Okay, or as in this picture, to illustrate all the combinations we could possibly have, we've got 8 haploid cells from 2. Okay, so hopefully that makes sense. That's basically where variety comes from in meiosis. Okay, now the maths, the numbers. Now this shouldn't be too difficult. Some people get this straight away if they're mathematically minded. Some people might need to see my wonderful PowerPoint animations here again. So let's go through this. For each chromosome then, with independent assortment, the probability of a new cell getting the maternal or the paternal is 50-50, right? So the new cell, when you get that independent segregation at meiosis 1, it's 50-50. You're either going to get all the maternal or all the paternal, okay? So half the cells, if we scale this up, half the cells are going to get the whole maternal chromosome and half of them are going to get the whole paternal chromosome. For this bit, we're kind of ignoring crossing over a little bit, right? Crossing over is obviously added to the diversity, but just to get the point here, we're kind of ignoring that for the moment, right? So really what could happen is that's one thing, right, that could possibly happen. So new cell 1 ends up with the maternal chromosome and new cell 2 ends up with the paternal chromosome, or it could go the other way, right? New cell 1 ends up with all the paternal and new cell 2 ends up with all the maternal. I hope you can see that is a 1 in 2 chance. The probability there is 1 in 2, or 50%. Okay? Now we have to amplify this up by more chromosomes. You don't just have one pair of chromosomes. You've got 23. Different animals have got different numbers, right? So, here's the possibilities that could happen with 2 chromosomes. Okay, so this is maybe chromosome 1 and chromosome 2 in you, the first 2 pairs of chromosomes. So they line up, they form their lovely tetrads, then maybe, all right, maybe, there we are, we're getting lagginess now, maybe new cell 1 gets both the maternal and new cell 2 gets both the paternal. Possible, okay? Maybe it goes the other way around. Maybe new cell 1 gets both the paternal and both the maternal. Also possible, okay? Or, because it is independent, random, maybe new cell 1 goes paternal, maternal, and new cell 2 goes maternal, paternal, or it goes the other way around as the last possible option, okay? New cell 1, maternal, paternal, and new cell 2, paternal, maternal, right? So basically we've got a 1 in 2 chance for each independent chromosome pair, right? Remember, independent assortment, okay? So if we put some numbers to that, right, for 2 chromosome pairs, right, it's a 1 in 2 chance each. That's 2 times 2, or 4 possible outcomes, or 2 to the 2. So 2 possible outcomes and the number of chromosome pairs, which is 2. So 2 to the 2, 4. Easy. Now, I couldn't be bothered to animate any more than this because you should be getting the idea. If we had 3, there'd still be a 1 in 2 chance each, but there'd be a 1 in 2 chance for the first pair, a 1 in 2 chance for the second pair, and a 1 in 2 chance for the third pair, and these are independent, okay? So that's 2 times 2 times 2, or 8 possible outcomes, or 2 to the 3. Now we could keep going with this all day, but it's easier to put n in, hence n and 2n, right? So for any organism that's got a base n number, for us it's 23, for other organisms it's different, flies it's 4, you know. For n chromosome pairs, each chromosome pair still has a 1 in 2 chance, but that's multiplied, or basically by the number of times, so 1 in 2 times however many we've got to the power there, right? So for us, that would mean it's 23 chromosome pairs, 1 in 2 chance each, so that's 2 times 2 times 2 times 2 times 2 times 2 times 2, 23 times, okay, or 2 to the power 23, okay? Now that's a number 8,388,608 possible combinations of chromosomes, and that's ignoring crossing over. Okay. Now this explains why two parents can have siblings that look very similar to each other, but they share some traits, but they'll never be exactly the same. Because with one parent producing that many different possibilities, and then you've got to multiply that by the other parent as well, who's also producing 8,388,608 possible combinations, right, of different different chromosomes, different mixtures in each gamete, and their random fertilization takes place, right, of one sperm and one egg. Well, that leads to a number of 7.04 times 10 to the 13 possible outcomes, and that's before we even talk about crossing over. Okay, that's just from independent assortment. Okay, and if you're not happy with that way of expressing numbers, 7.04 times 10 to the 13 or 704 with 11 zeros after it, if you're not happy with standard form, why are you watching this video? Right? What are you doing at A-level standard? Be okay with standard form. Okay, so that is why we end up with such variation, okay, at meiosis. Okay, you've got the crossing over, which gives you new combinations of alleles, and then you've got independent assortment, independent segregation that splits the chromosomes up in random different patterns. Okay, so in summary, right, variety is created by crossing over at prophase 1, okay, that's one place, and independent assortment at metaphase 1. Okay, so metaphase into anaphase 1, right, that's independent assortment. Okay, and then you have to multiply that by the fact you've got two separate parents at fertilization. Right? So this leads to the fact that there are so many combinations, right, that you are completely unique. Right? You are the only person who has ever existed and probably ever will exist with your particular combination of chromosomes and alleles. Okay? There is nobody else on this planet who has lived or will ever live with exactly the same DNA as you. And I include genetically identical twins in that, monozygotic twins, because there are slight differences in the DNA, turns out, between identical twins. Obviously they're a lot closer than the rest of us, right? Pretty much identical, but not fully. Okay? So that's obviously quite a lovely thought to think we're all completely unique, right? We're all special creations, of our parents, of completely random mixtures of all the genes that have gone before us. However, when you put these differences in context, I mean, we've all got arms, we've all got legs, and the liver and our cells work in the same way. So this is a quote here from an article in Nature called A Global Reference for Human Genetic Variation, which says the typical difference between an individual's genome and the reference genome was estimated at 20 million base pairs. Sounds like a lot of base pairs, but actually when you compare that to 3.2 billion base pairs, that's our full genome, that's only 0.6 of the total. So you are actually 99.4-ish% the same as the person next to you. Now obviously if they're a relative, they're going to be more similar, and if there's someone from a completely different ethnic group from another side of the planet with a completely different heritage and lineage, then they're going to be a bit more different, but on average. So I'm saying you're completely unique, and that is true, but the differences, they only make up about 0.6 of your genome. So always remember, and this is where we're going to finish, right, that you are completely unique, right, just like everybody else. Okay, many thanks, and I'll see you next time.